I want to thank Agilent for having me here today. I'm here from UCSF Dermatology, and we've been working together for a few years on using deep sequencing and exome capture to try and understand human cancers better. And so I think we have some pretty interesting ideas and approaches to talk to you about today. I think some of you are probably asking what the connection between dermatology and cancer genetics or exome capture might be. So I wanted to go over a little how we think about skin cancers. They're really a remarkable model for a large class of human malignancies. So if you take cutaneous SCCS, they're really a prototype for epithelial cancers, as I'm sure some of you know. And if you lump them together with lung, esophageal, cervical, and head and neck SCCs, to which they're very closely histopathologically related, that group is one of the most common and deadly of all human cancers. And obviously, being an epithelial cancer, they also have things to tell us about other epithelial carcinomas like breast cancer and prostate and colon cancer. We see about 1 million cases of SCCs of the skin in the United States, just largely because of sun damage. And these are really easily diagnosed and treated for the most part, but because there's so many, inevitably some of them become bad actors. So we still see about 2,000 or 3,000 deaths a year from cutaneous SCCs. The treatment is largely surgical, so even for a basic scientist like me, I end up cutting out a few of these in clinic every week. And the standard of care for resistant disease for a long time, for kind of metastatic disease, has been EGFR inhibition, but it's been pretty dismal in terms of response. And I think, as we all know now, the incidence of amplification or mutation for EGFR is remarkably low in these cancers. And obviously, dermatologists also look at melanomas, which are a prototype for neural cancers, and we use them to understand specific subsets of melanoma, for example, uveal melanomas, which occur in the eye, and other neurally based cancers like neuroblastoma, about 60,000 cases a year, far fewer, but as you all know, much more deadly, with about 10,000 deaths a year. Treatment for melanomas has been surgical largely until the past year, when vemurafenib, the first inhibitor of the driver gene BRAF, which is mutated in about 50% of melanomas, came online. So that's been a huge sea change. And recently, immunoadjuvant treatments for the first time have shown survival increases in melanoma too. And already, I think it's been shown, I think probably as at least some of you are aware, that in cases that become quickly resistant to BRAF inhibition, that inhibition of the parallel MEK pathway appears to offer even further survival benefits by overcoming resistance. So skin cancers have a lot to teach us, but I guess really my point today is this isn't even the biggest reason to get excited about skin cancers. That the really big deal for us and the way we think about these cancers is, let's move this off, is that because of UV radiation, skin cancers acquire 100,000 or more mutations per genome. And this is in contrast to just a few thousand mutations in solid cancers like breast or prostate cancer. And this has been of great interest to us because we've been using, and there are a number of groups that are starting to do this now, and are actually, I should say, are very well along, and it's becoming a focal point, using mutations in genomes to recreate an evolutionary history, kind of an archaeological record of what happens to these cancers as they become more clinically devastating. So in a sense, we're trying to take those huge numbers of mutations and really trying to understand which ones came first and then which ones show up later as the cancer evolves. And kind of the methods that we use and some of the technical methods and the thinking that we use to unravel this is what my brief talk will be about today. Obviously, we didn't always think that cancer was going to be this complicated. Just 6 years ago, which seems like 100 years ago now, there was still this model that you went very simply where a normal cell would accumulate progressive numbers of mutations, each of which would enforce transition to a more malignant stage, and we'd be able to just find a small number of drivers we could inhibit, and we'd be able to get a curative response. And obviously today in 2012, that seems like kind of a fantasy, and cancer has turned out to be challenging genetically for about 100 different reasons that are interesting. The thing I basically wanted to focus on today is this idea that certain mutations to promote tumorigenesis actually need to happen in the context of other mutations that happen first. You know, if you put p53 back into a cancer that's lost it, you're not going to see much of a response. So there are really sequences in which these aberrations have to be acquired by cancers to come up with a malignancy. And so what this means is it's not enough just to understand all the genes that are mutated in cancer. You also have to understand something about which ones are important at which phases. And this is really the question we try to focus in on when we start doing this work with these technologies about 2 years ago. So the question we wanted to ask had to do with premalignant mutations and what happened early in the process, because especially in the skin, precursor lesions are identifiable for cutaneous SCCS, which is what I'm focusing on today. We got about a dozen cutaneous SCCs and matched normal tissue from our UCSF dermatology clinics. These were sequenced, I think this was back Agilent 2.0, the SureSelect kits. So we went for whole exome sequencing at about 50x median depth, which now does not seem very deep at all, but back then seemed extraordinarily deep to us. And as I was telling you about, what you actually see is about 1,200 to 1,500 mutations per exome, and this translates to north of 100,000 mutations per genome. So we use this to kind of assess these mutations in the context of chromosomal aberrations, which I know is an interest of the group today. So I just want to take a few seconds and really explain how we think about this. This very, very simple example relies on a specific abnormality known as copy-neutral loss of heterozygosity or copy-neutral LOH, which I think is probably familiar to many of you. And the idea is that you have a diploid state and you have duplication of one chromosome or one part of one chromosome and then loss of the other. So you basically have, you preserve a diploid state, but now you have two copies of one chromosome and you've completely lost the other. Well, it turns out that in cancers that are heavily, heavily mutated, like skin cancers, what that means is the chromosome that is duplicated, all the mutations that were gathered along that chromosome double in copy number. So essentially, early mutations now acquire, after this copy-neutral LOH event, a copy number of 2, whereas all the other mutations that happen after this event happen in single copy number and persist in single copy number until the cancer's cut out. So the theory behind this is that you basically have a copy number of 2 in earlier mutations when you look at regions of this kind of specific duplication. And when we went to look at the actual data, turned out to play out really beautifully. So I'm just going to acquaint you with this diagram here. This is one chromosome 17 in a single SCC that we sequenced. Along the x-axis here, we have the genomic position in megabases. And along the y-axis here, we have the allele frequency. So this is after exome capture and sequencing, the actual allele frequency of the actual mutations. And the colored dots along here represent mutations that were identified through that exome sequencing. And as you can imagine, if you have, in a normal situation, a mutation on one chromosome and obviously not one on the other, then you'd have an allele frequency of about 0.5 is what you'd expect for this line, because there's some contamination with normal tissue that gets compressed down, but you can see a very nice theoretical heterozygote mutant allele frequency. In this region here, you actually have a region of copy-neutral LOH. And then you can see kind of lonely up here, we have p53 being mutated at roughly the expectation for a homozygous mutation. And if you think about this long enough, which we did, the only way you could come up with a situation exactly like this is that you had one mutation in p53, and then the genome remained clean of mutation for basically the entire life of the cancer until that chromosome duplicated and got rid of the second copy of p53. If you even had a few mutations on that arm before the duplication, those would have gotten doubled in copy number 2. But you can see that it's really, really clean here, and it's clean in virtually every other case we've looked at in SCCs and ovarian cancer. So what this means biologically is pretty interesting. It means that even after a single p53 mutation, the genome remains almost pristine of mutation. And this is, I think, as you know, in contrast to the classic biochemical model of p53, where one mutation is enough to kind of poison the complex, that for purposes of keeping the genome clean of mutation, the p53 is effectively very, very recessive. The other thing this suggests is that p53 mutation comes very, very early in the process of the genome getting progressively strange or mutated. And this is in contrast to the classic Vogelstein model 20, 30 years ago, looking at colon cancers and deciding that p53 mutation happens late after there's been a driver mutation that pushed a cancer towards senescence. So we actually then tried to use this to order not just mutations relative to one another, but also chromosomal duplications. So let me just tell you, you can use some of the same thinking here. So if you have a duplication that occurs very, very early, what that means is all the mutations along that region acquire a copy number of 2, like we were talking about before. But now that region has a long time before the cancer is cut out to acquire additional single copy mutations. If you have a duplication that happens much later, let's say shortly before that cancer is cut out, then it has a shorter time to acquire single copy mutations. And according to this model, the idea is that the earlier duplications are more heavily populated by single copy mutations. And again, when we go to the data, we see something similar. So this is chromosome 17. This is one we were showing you before. But hopefully now you're looking at this duplication and thinking, wow, that's really predominated by single copy mutations. And so that's what we think of as an early duplication in the life of this cancer. And then if we now look at other duplicated regions marked in here by this pink, you can actually see that there are certain ones that, this has a lot of single copy mutations, but they're now closer in proportion to the homozygous mutations. So we think of this as kind of a mid-course duplication. And then you can see this duplication here, the CNLOH event, that's predominated by homozygous mutations. And so we think of that as having happened very late in the lifetime of that cancer and not having had a lot of time to acquire additional single copy mutations. So this is a way of being able to order these in real time. These are statistically separable. These methods have been advanced since we published this paper. There's a statistical group we've been working with Elizabeth Purdom at Berkeley, who's been trying to extend these approaches to cytogenetic abnormalities beyond simple CNLOH, which is obviously for many reasons the easiest to look at. What this tells us biologically is kind of interesting. It tells us that p53 not only seems to precede the vast majority of the complete loss of p53 is required not only for the vast majority of simple mutations in the genome, but also for virtually every chromosomal abnormality we could find in this cancer class. Okay, so I'm gonna wrap up now just telling you a little about how that also now provides a larger context. One of the things we also found in that study was the fact that Notch1 is a tumor suppressor that's mutated very commonly in cutaneous SCCs. So that was our finding here. And in the past year, it's come out as kind of a pan-SCC tumor suppressor in lung SCCs and head and neck SCCs and now esophageal SCCs. So this Nature Review in Clinical Oncology kind of encapsulates the growing situation, becoming a major target in terms of pathways for pharmaceuticals looking at this malignancy. These are clearly disabling mutations. They're truncation mutations that really disrupt protein structure. And they happen in 70 or 80% of cutaneous SCCs. I mean, that's a tumor suppressor frequency that, as you know, if you follow cancer sequencing, you just don't see with genes other than p53. So we expect this to be a pretty central gene in squamous carcinogenesis. And the reason I just wanted to bring this up, I want to just take about 2 seconds to tell you about the biology. What Notch does is it's a cell surface receptor, but the ligand that it binds is actually bound to neighboring cells. So it kind of enforces the cell-to-cell contact, and when you get that contact and ligand engagement, the signaling causes a higher degree of differentiation in epithelial cells. So by losing that signal, you get more invasive behavior, de-differentiation of these cells. And so one thing we noticed using this timing method here is that these mutations reliably appear to occur after p53, probably about 3 examples out of 6, where the homozygous loss of Notch happens after the complete loss of p53. So now we're able to kind of order these with p53 happening earlier and starting to create this kind of fertile soil for heavier mutation, and then mutations, at least like Notch, and there are probably going to be others that come along, that make the cell less differentiated and start exhibiting more malignant behavior. So it's a way to really integrate all these different observations on the sequencing level to give us a timeline of how these cancers progress. So that's pretty much what I want to tell you about. Hopefully we're kind of using the same techniques now in melanoma, kind of a very, very large series, so hopefully we'll be able to talk about that to you at some time. But it's been our ability to look at this number of samples at each stage when the sequencing volume was severely limited wouldn't have been possible if we hadn't been able to access technologies that were this robust. Thank you.